Good morning, everybody. I think it is time to begin. First off, to kick off the entire event, I would like to invite up Dr. Jennifer Roberts. She's the director of the Resilient Systems Office at ARPA-H and has really been foundational in pulling together a brand new office amid all of the hustle and bustle of our first year. So, Jen, want to introduce ARPA-H?
Thanks, Paul. Great. Good morning, everybody. Great to have you here and great to be in Denver. So I wanted to start off by talking a little bit about ARPA-H, give an overview of the agency, and then I'll hand it off to Paul to talk more about the main event for today. So the ARPA-H is a brand new startup organization inside the government, which is a very unique place to be. And our mission is to accelerate improvement of health outcomes for everyone. So there's a couple different parts of our mission. The accelerate part hits on the fact that we are interested in breakthrough technology research. We want to have things that are revolutionary in nature and things that are really trying to push the envelope on what's possible. We're interested in health outcomes. So not just things that are working in the lab, we want to be able to translate that into technologies or approaches that will help people in the real world. And the for everyone indicates that we're not just interested in making progress toward new types of interventions that may not exist today. We're also interested in taking things that already exist and making it so that they can reach the patient populations who need them. So perhaps there's something where there's a capability available in an academic medical center, and we want to make that available to folks in a home health setting, or we want to bring down the cost of a therapy that costs like $2 million per dose so that it can reach the patient population who needs it most. We were started recently by the current administration, and I like to include this quote talking about pursuing ideas that break the mold on how we normally support fundamental research and commercial products in this country. So effectively what that means is ARPA-H is interested in funding types of research that are not well supported by other partners or other funding sources in the ecosystem. So for example, we have wonderful partners at NIH and other organizations that fund research that really expands our understanding of the science in very, very important ways. If something is already well-funded under those models, NIH, NSF, other types of models, then it's not the area that we're looking to be in. We're looking to address the gaps that are not filled by those other sources. Similarly, with the venture capital ecosystems, there's a very vibrant venture capital ecosystem. There's also a very vibrant ecosystem that includes lots of large companies that invest in new innovation. If something's well supported by those parts of the ecosystem, that's also not the space that we're trying to be in. We're identifying where are the gaps, where are the things that are not well supported by those other methods of funding. As an organization, we're unique in that we are an independent agency inside of NIH with our director reporting directly to the secretary. So that's very unique to us, and it speaks to the different type of mission and business model that our organization has. As we're standing up, we are building a network of organizations and communities that will both help develop the technologies that we are aiming to create and help test them in the real world and ultimately provide a path toward long-term use and sustainment. So we have our customers, the folks who are patients, healthcare providers, the public. We have the performers, which then academia, industry, small and large businesses, nonprofits, and then a variety of other partners that include private investors, FDA, our regulatory partners, who can help create a path toward whatever technologies coming out of ARPA-H program living on in the real world past the end of the funding that we provide. So I'm the director of the Resilient Systems Office, as Paul mentioned, and our office is interested in making it so that we have a more integrated health system. and looking at new ways to make it so that the system is more robust and resilient, both to make it so that high quality care is easier to access and more available, and also to make it so that if there are unexpected disruptions, pandemic comes to folks' mind, but other things as well, workforce shortages, cyber attacks, et cetera, that the system is well set up to be able to navigate through and continue to operate effectively, even when there is some type of stress put on the system. So we're interested in a variety of large revolutionary type challenges. So you'll be hearing more from Paul today about program on how to think about achieving the body making its own medicine, right? How does that fit into a resilient system framework? Well, if the body can make its own medicine, that makes it so that patients need to visit the doctors less, right? It makes it so that we can have a much more distributed way of producing medicine and reduce the burden on folks with chronic conditions. And then to give another example of a program that will be doing a proposers day tomorrow. What if the body, or what if we could eliminate viruses as a class, right? So instead of having a vaccine for every single virus, we had vaccines that worked for whole classes of viruses. So what if there was a single virus that made it, or vaccine rather, that made it so that you never got the flu again? That type of thing. Our business model is unique. So ARPA organizations are centered around program managers. And the way that this works is a program manager with a cool, innovative idea comes to ARPA-H and stays with us for a tour of 3 to 6 years. And during that time, they will develop a series of programs that tend to last somewhere between 3 and 5 years. And what this does is a couple things. So we are constantly pulling in folks with new ideas who are at the cutting edge, and that helps us remain agile as an organization. It makes it so that we can keep on updating the portfolio with great new concepts by pulling in folks that have a different perspective. And then that provides us with the ability to do a series of programs that really are all trying to push the envelope in different ways. So what you'll see in the next 2 days is really some very fundamentally different programs. One's about the body making its own medicine. The other one's about universal vaccines, right? And what you can expect to see from ARPA-H is a steady stream of programs that will each focus on a novel problem that could have a revolutionary impact. In terms of the way that the program formulation process works, so the program managers first develop a concept, they refine that inside of the support system inside of ARPA-H in order to be able to turn that into the type of solicitation that we'll be talking about today. We'll launch the program and then we'll select a set of performers to be able to go and execute. And we're really looking for a variety of different approaches so that we can have a balanced portfolio to maximize the chances that the program vision can be successful. And then during the program, we have folks try a bunch of different ideas about how to achieve that program vision. Some of them will work, some of them won't. This slide does show a type of formulation where one performer comes up with a solution. Honestly, what I find is more common is that a lot of different performers will find different parts of the solution. And we end up with a hybrid approach that takes the best of everybody's techniques. And at the end of the day, what we're looking for is that technique that is providing the most value, the clearest chance of success in real-world application. And at the end of the program, we will work to try and provide a pathway toward that technology reaching the patient population or the population of clinicians or other stakeholders that it needs to reach. And with that, I'll hand it over to Paul to get to the main event.
Thanks, Jay. All right. Once again, good morning. Thank you all for showing up today. I just want to make a point before anything else. It's called Proposers Day because it's for you, right? Proposers Day is for the proposers. It is not ARPA-H Day. It's not Program Manager Day. Today is for the proposers to fully understand what we're asking for, right? So I have spent many years on your side of the table asking for money. I know the worst thing in the world is a bring me a rock kind of proposal. And, you find out if it's the right rock. Today is about explaining as fully as we can what we're looking for, the scope of the ambition that we're looking for, any technical challenges, all of that should be clearly defined. So we have lightning talks for you, we have poster sessions for you, we have sidebars, we have opportunities for you to talk amongst yourselves, to form teams. We're open to other suggestions, right? All when we put together this day, it was all trying to establish, different times of the day for you to think and organize and team in different ways. If you have a brand new idea, we're open to that for the next. Okay. All right. So today we're having a proposal stage for resilient, extended, and automated cellular therapies. So one of the things I want to foot stomp about ARPA-H. Many of you are familiar with DARPA, where I came from. It's a fantastic place to work. It's often like, wow, can we even just pull the following thing off? And of course, you want to translate it. ARPA-H is about translation. So translation is about translation, right? And that means all the way to the patient. You have to think about how is the patient going to live and experience this therapy. So whatever thoughts, concepts, technology that you're thinking about, what would that be like in the patient's hands? Okay, so I just keep that clear in your mind. And so the big question here is like, what if we could provide affordable therapeutics to help patients manage their health regardless of who they are, where they live, or right, and what their socioeconomic status is. To do that, we're building out what's say is two devices that allow you to connect with the body, right? So the first, I call it living pharmacy. This is a bioelectronic device that controls cellular factors, right? So part of it is the bioelectronics that connects to cellular factories. It communicates with those cellular factories electrogenetically, optogenetically. There's a large variety of techniques for these bioelectronics to control the cell therapies. That's all encapsulated and implanted inside the body, right? And so that allows you to stimulate the cells to deliver a range of therapies when and where is needed, that are needed, and to do so automatically, right? So it controls the cellular factories or have a controlled release of the therapies. We have many different possible applications, right? If you just actually take a step back and you think about pharmacology, like 2/3 of pharmacology are going after hormone receptors and cytokine receptors, right? So this is, you can imagine there's a broad range. So we throw out different examples to give people ideas. I mean, diabetes, obesity are obvious targets, but we're also interested in thyroid disease, a broad range. And so part of your job in the proposal is coming up, right? There's a brief 1 page. What is your problem? Do you have a large enough patient population to actually test it out? How are you going to deliver the therapy? What is that therapy? What is that going to look like for the patient? The other devices is a bit of a mirror image, but many of the main components are similar, right? So again, it's a bioelectronic device. Again, it contains engineered cells, but here the cells are sentinels, right? They are monitoring what is happening in the body, and they produce a signal, right? That could be a range of signals. It could be a fluorescent protein. It could change it by electric potential of the membrane, bubbles, what have you, something detectable by those bioelectronics, and those bioelectronics then export that information outside of the body back to the patient, right? And again, you can think of many things that one would want to track on chronic inflammation, obesity, Crohn's disease, et cetera, right? So 2 different devices that again allow the patient to control what's inside their body and to understand what the disease state is inside of their body, okay? So why are we doing this, right? One of the main reasons is to focus on chronic diseases, relieving the burden of managing therapy. And this is one of the things that stunned me. What an enormous problem this is. And the example I tend to throw out is like for these, many people have heard of all these GLP-1 agonists like Wegovy that are, so popular, have such a profound effect, right? So one of the, what you'd think would be a really easy thing with these is like you only have to give yourself an injection once a week, right? It has a huge effect, like a huge weight loss, and of course, a drop in weight has great benefits for long-term health. And so, after a year, the compliance, the people who are still in that therapy, is down by half, right? After 2 years, it's at a third. And so you think, wow, like how much easier can we make this one fairly easy injection per week, right? And so that's the impetus. Clearly, we have to continue to push on reducing the burden on the patients, right? So you have just some of the numbers, like many people at some point in their lives will be faced with this lifelong burden. 60% of adults have one or more chronic diseases. Up to half the patients are non-adherent with the medication. Right? You can imagine, like, oh, we come up with all this great science medication, and then if it's not easy for the patients, it's not going to be used, right? And then easily considered a preventable cost from $100 to $300 billion each and every year, right? There are a lot of reasons for treatment non-adherence. Cost and not lacking insurance are part of it. One of the themes in this is trying to drive down the cost of these implants. Sometimes it's treatment aversion. People don't want to give themselves shots or pills all the time. Optimal dosing time is often a challenge. Like, you have to remember to take it right after you eat or before bedtime. Here, you can imagine with a device that's implanted, therapy can deliver it automatically, and you wouldn't have to constantly remember. So, and you also see forgetfulness, or even, in elderly populations or a variety of populations, sort of unclear instructions. So the goal of REACT, right, we want to create these implantable devices that use these cellular factories to produce and precisely deliver the therapies that can track disease or biomarkers, are modular and tunable, right? And so this is one of the arguments and how we're going to drive down the cost, and we'll get to that in a bit. And ultimately relieve the burden of managing therapy so that we can improve patient health, right? So how is this going to work in practice? We're trying to go from what we'll call a prescription model of therapy to subscription, right? So you go to your doctor and you hear the unfortunate news that for the rest of your life, you're going to have to take 2 pills twice a day. So now, instead of constantly thinking about renewing prescriptions and getting yourself a pill organizer or whatever it takes for you to get it done, instead they propose having a small implantable device. The prescription on the clinician side is they set a range for those therapies. You then go and with a small minor outpatient procedure, you get this implantation. Maybe it's in the upper arm, maybe it's in the abdomen. That's part of that is on you to propose the optimal placement. But you have a small implantation. And then what the patient needs to do is to subscribe to the therapy, right? It's very important that the patient have autonomy. They're in control of this. But at the same time, there's not this chronic burden on them, right? What I don't want is every time they need an injection, they have to press a button, right? We haven't solved anything. It should automatically produce the therapy where and when it's needed. So once they subscribe, the therapy begins. Then they have medication. The cells secrete the therapeutic biomolecules. If needed, or even separately, there's tracking, right? There's that separate living sentinel device, also implanted, that can track what's going on in the body, so the patient can track their therapy. And then ultimately, they get confirmation either that the therapy has been released, or they know what they're, for the living sentinel, that they're within the parameters as defined by the clinician. Clear enough? Awesome. Okay. So I think there are many benefits of this. I think it combines the best of multiple current therapeutics, right? So there are precedents, right? There are these implantable pain pumps, right? So you get accurate patient control over the therapy. You can think of these artificial pancreas type approaches, things like the Omnipod. And please note here, I'm going to give specific examples because I think you need, to know what inspires us. None of these limits what you should propose, right? But I'm just trying to give an image of what I'm thinking about, right? So these Omnipods are pretty cool, right? Continuous biomarker monitoring to track the state of disease. Great work by Vertex, other companies implanting beta cells for continuous production of native molecules, right? So we're trying to pull all of this together into one device. All right, so what needs to be done to make this happen? We have multiple TAs. Normally in ARPA programs, there's like three TAs. I'm trying to break this up into several of them so that each and every one of them is distinct. So one is long-term maintenance of cells in vivo. Right? So on this first pass, we are trying to get to stable delivery for at least 1 year in vivo. Right? So you're taking cells. Maybe you're planting them in a milieu inside the body that is not native, right? Maybe they're, the beta cells and you're implanting them subdermally. It's a different area. How do you stabilize that phenotype? Is it going to drift because it's seeing other soluble factors in vivo? How do you keep that? Do you add, helper cells? Do you change the mechanics of the implant? How do you keep those cells stable for extended period of time? The second one is improving the manufacture of standardized cell lines. And I think this actually captures, sort of the difference between the DARPA and the ARPA-H thing. And DARPA was like, okay, let's just get it done. ARPA-H, we want this to be low cost, right? We want it to be manufacturable. And so we are requiring people to think about how these are going to be manufactured. We're requiring the proposers to come up with new processes, right, to engineer cells to manufacture them that is going to be commercializable, right? And we're specifically opening that up so that the resources, so that you can think about that as you're developing them. So TA3, the third task is this implantable device that communicates with the patients and with their doctors. And then we split up a little bit. There's TA4 and TA5. TA4 is the living pharmacy. It's the therapy generation with stimulated release, making sure that that is reproducible, accurate, trackable. And then TA5 is this accurate biomarker detection. Okay, again, trying to give some examples of how this looks. Examples, you have an external device, a smartphone, other device. You have that can communicate with the implanted device. It communicates, potentially recharges of that device. You have in the device, you need the accurate delivery. And if you can think of the architecture here, you have some membrane that's surrounding the entire device. You have some way of communicating the signals. Here we have electrodes. Maybe you have helper cells that help the other cells stabilize. Maybe you need to be able to isolate this device from the immune system. Other coatings, cells like you may need to simulate a basement membrane to stabilize your cells, right? So all of these are the types of components that would go into a living pharmacy or a living sentinel. So the general timeline is we have 3 phases of the program, 3 main steps, right? And so the first is development. We want you to develop all the different components with an eye towards eventual integration, but show that you can pull off the metrics, the performance in each one of these task areas. The second phase is you're going to take all these components and we're going to integrate them and begin to test them. And then the third is a phase 1 clinical trial, right? So those of you who are familiar, you need to know fairly early on how these pieces are going to work together in order to get to that phase 1 clinical trial. So what I've put up here are what I call key metrics and milestones. So the ARPA model is very milestone driven. We ask for you to pull off crazy things, right? And then at the beginning, we set these markers, right? So if at the end of phase 1, you can really pull off this, we're on the right track. And that's just sort of a marker for you, for us, so that we don't, go, well, this is kind of cool, but we've got to be honest with ourselves. Are we actually on track? So those are the milestones. These are the key ones. You'll see that each one of the TAs have other ones, but these are, I would say, less negotiable than the other ones. There's a variety of techniques that you guys, clever ideas that you guys are going to come up with. And so for other TAs, we might go, okay, maybe, we can bend this way and that way, but you should really think about these, right? So at the end of, in phase 1, there's a midterm task. Well, after a couple of years, you need physiologically relevant secretion rates from encapsulated cells in vivo, right? So it actually needs to produce. That doesn't mean long term, but at a certain point in time, you need to have enough cells in a reasonable form factor in vivo. For the living sentinels, the limit of detection in vivo has to be at least 10 times higher than the therapeutic range, right? So it can be at a higher concentration, but you need to be on track. You need to be within striking distance of the ultimate limit of detection. And that's for whatever, chosen biomarkers that you have in spiked clinical samples. By the end of phase one, the delivered therapy improved the animal model's physiology, right, after 6 weeks. So there's a little bit of durability of the cell lines here and also a functional impact. But for the living sentinels, the limit of detection should be approximate to the therapeutic range for the chosen biomarker in vitro. Into phase two, again, in phase two, we're pulling things together, show that the delivered therapies improve the large animal model's physiology after 1 year, and then the sensors are accurate for 1 year in vivo, right? And again, next step is the phase one clinical trial. A different set of metrics to get the same endpoints, right? So these are important, but they're not as key or principal as the metrics I was just talking about, right? So again, to give you examples of some of the metrics you might see, you need to establish after a year the biocompatibility of an optimized immunoisolation membrane or coating tested for at least 4 weeks. After 24 months, you need to demonstrate in vivo viability of at least 70% after 2 weeks. After 3 years, 7%, and all these are in the DA, 7% viability with at least half of the viable cells having maintained function after 8 weeks, right? And so that keeps on escalating, right? And the rationale is that you're implanting cells, right? If they all die, it's going to be very hard to recover. If they lose their function, it's very hard to recover, right? So our estimation here is that if you have at least 70% viability and 70% function in those cells, right, then you've lost about half your generation capability. So our estimation is that you could probably push the cells a little bit harder, doubling their output, but you push them any harder, they might burn out. You may have different ideas, right? So you can come up with different metrics to show that you have these long-term viability, right? So if you have amazingly productive cells and you can get it done with even with 90% die-off, that is up to you to convince us that you have the technologies that can pull that off. Again, this is not limiting the technologies. You may have other ideas. These are just some technologies that we have seen that makes us feel that this is going to be possible, right? So a couple of things off of bioRxiv. One of the issues, you plant cells inside the body. Oftentimes, they're used to growing externally within high oxygen content, right? And that low levels of oxygen lead to low levels of productivity. Here, recent work from O'Connor out of Sahib Cohen-Carney's group, local oxygen production, right? So splitting water in vivo to maintain the performance of the cells, greatly increase the viability after extended implantation, right? Isolation membranes, people have been working on this as well. They're in clinical trials. They're getting better and better as measured by the amount of fibrosis around these, right? So I think that technology is great. People continue to improve it. What I think is going to really polish it and get over the line is people developing combinatorial techniques for testing materials in vivo. So an example, another bioRxiv paper out of the group of Calder Forest, sort of high throughput screening in vivo, where they come up with a large combinatorial matrix of materials that are implanted at once, right? Very cool. You can do, 49, I think that was in the murine model, many more, and in a porcine model, all at once. Of course, greatly reduces the number of animal testings while expediting testing, right? So these are the kinds of things that we're looking for. I'm sure you have even more clever ideas. The second example is improving the manufacture of standardized cell lines. So I would say a fairly significant choice in this program is that we're going after allogeneic cell lines, right? I have just not seen any technology that's really, really going to bring down the cost of autologous cell lines. So the idea is to have an allogeneic cell line, engineer it, have stable cell banks, and then use the encapsulation to deal with immunogenicity. So here the goal is to develop, commercializable commercial cell lines that lower the entry cost for treating new diseases. Metrics, here, the manufacturing it's sold, some with the optimal chassis, screening different kinds of cell types. What's the optimal one, right? If you in a lab and you're, oftentimes someone has their favorite cell line. Here, we're giving you the time, the resources that you can go after other cell lines, analyzing that particular cell line, finding safe harbor sites for stable integration, and then sort of really pushing on that. There are other things that one might propose. We will hear later from Army about some of the capabilities that they bring. Anything that you want to bring to bear on this and how it might improve the manufacturability of these cell lines, we're open to hearing. Right? So many people have proposed different technologies out here. Examples of a recent paper by Suresh. So I should know that, basically analyzing the genome, finding these stable sites that don't lead to cancer, but also lead to that are also less likely to be silenced and that are stable. And people have pushed really hard on this, coming up with systems that can screen, bacteria and yeast, screen large libraries all at once. We're open to beginning that process for mammalian cells, coming up with new ways for rapid screening and optimization of mammalian cells. So that is to you to propose. So implantable devices that communicate with patients. So the goal here is to engineer, the transduction and the communication capabilities so that you have continued performance for about a week, right? And so we say a week in that we don't want the patient to have to charge up their device, 3 times a day, right? And so our estimation is about once a week is a good breaking point between you want the patient to know that they have the device, take care of the device, but it's not a burden, right? So we're open to other concepts to how to recharge, reload, communicate with the device that minimizes the patient's cognitive burden of maintaining it. But for here, the sort of baseline goal is for at least 1 week. So you have to demonstrate the sort of optimization of the transduction mode in vivo, making sure it's energy efficient. At 36 months, demonstrate in vivo testing with the cells for at least 6 months. And after 4 years, demonstrate that the system can function on a single charge for 1 week in vivo with reserves, right? We don't want it to just die after 1 week. It works for 1 week, but then if you're not charging it up, then it'll you can still communicate with it for another week. All right, so the key enabling technologies here, and I think this is a bit of the fulcrum of the proposal, is figuring out how you're going to engineer the cells, right? And so there are many different, this is a great review that came out a couple of years ago. There's a wide variety of techniques that people have developed that can turn on the production of a gene, electrogenetic approaches, optogenetic approaches, mechanogenetic approaches, even magnetogenetic approaches. And the concept here is, I think this is the fulcrum, right? Because at some point you have to decide on this, right? And the synthetic biology people are going to have to engineer the cells, and the electrical engineers are going to have to engineer the device, right? And so everything sort of, splits at this point. You have to really determine early on what is the optimal transduction method, what are the benefits, what are the weaknesses, why you're choosing this particular mode, right? So I put this here because I think it's a key choice during the development. So within the program, performers will choose a track. There's the living pharmacy track, where again, for both of these, you go through task areas 1 through 3. And the living pharmacy is really going to focus on TA4 therapy generation with stimulated release. And then the living sentinel is going to have the accurate biomarker detection. So for therapy generation with stimulated release, so the goal here is to efficiently deliver between 80% to 112% of the target dose of the therapy in vivo. So that is sort of a baseline FDA requirement, 4/5 and 5/4. If you're proposing a cytokine, a therapy, a hormone, whatever, that has a tighter therapeutic index, then of course you have to take that into account, right? So this is sort of a baseline. How much control do you have over the therapy that's being delivered? So as you step through, we know that people in synthetic biology, they have lots of tips and tricks. You have to work through this. With the baseline after 12 months, you have to be within 10% of the production rate of the therapeutic molecule, at least in vitro, right? And so if you're trying to produce molecule X and you're only getting, 1,000th or 100th of it, my experience is you're not going to get there, right? It's probably you've chosen the wrong cell line, the wrong target, something's going wrong. So you have to be at least within striking distance after a month. Month 18, demonstrating modeling or animal studies confirming target dose of therapy to deliver in vivo. Concept growth is just PK/PD. Like if I'm producing a molecule that is normally produced in the brain and it is being delivered from subcutaneously inside the arm, you're going to have to work out the PK/PD so you ensure that it's getting to the right place, right? And there are a variety of modeling techniques that you can use, or you can use animal studies. After 2 years, demonstrate physiologically relevant production rate with an on-off ratio of at least greater than 10 in vivo, right? So it's up to you to define what is the physiologically relevant rate, show us the papers, show the result, what is the amount of production that you need to produce for your particular system. And then last, after 4 years, you have to confirm that PK/PD via device in vivo. A lot of different technologies that could enable this. I presume most of you are familiar with the SynBio side. I just want to mention a couple other techniques. One of the things you have to do is confirm that you're actually releasing the therapy, right? You imagine you're a patient, you get something implanted you. If you take a pill, you're taking your medicine. How do you know that it's working, right? So you need some way to confirm to the patient that therapy is being delivered. That is up to you to propose. Example I'm just throwing out here is that you can imagine for every 10 molecules of a hormone, you're going to produce a fluorescent protein, and then you're going to use, this is work by John Rogers, making these miniaturized spectrometer, right? And you're going to track that molecule in vivo. Modeling PK/PD, again, this can be done in animals if you want. However, we're also completely open to using one of the different modeling approaches that are out there. And then sometimes faster release. One of the things I want to remind you, some hormones, some therapies need to be delivered very quickly, say insulin, some very slowly, like thyroid. You need to make sure that you can match those kinetics, right? And so I just give an example here of sort of fast kinetics. This is out of the Fusenegger group of sort of really fast release. They engineered these electrogenetic triggers to release vesicles, right? And so you don't have to actually, to translate. You can just release vesicles of prepared molecules, and that can actually match those kinetics. Okay, so next for TA5, accurate biomarker detection. So here, the cells, again, are the sentinels, accurately transduce biomarkers at physiologically relevant levels in vivo, stepping through. Again, it's sort of a step through. This is all in the BAA or the NAI. We're trying to step through, making sure that you have the appropriate limit of detection in your cell lines, that is transduced by the bioelectronics, making sure, this is on you, that the measurements are at the Nyquist frequency or whatever is required to accurately track that physiological process. By the end of 54 months, you can demonstrate detection remains accurate in vivo for 3 months. And then final at the end is at least for 1 year. Lots of different technologies in this area, the SynBio side. I'm just, again, sketching some of them. This was actually the first one worked in a theranostic cell, sort of broad spectrum fatty acids, right? So for your disease state, you can tell me it didn't have to be just one molecule. It could be, broadly, we want to monitor fatty acids in vivo to track obesity, right? For instance. So that's broad spectrum. It could be a generic approach, sort of like the SynNotch technology developed by Wendell Lim. That's a very, you have that sort of notch core, and then you, on either side, intracellular, you have a transcription factor that can turn on whatever gene that you want, and externally you have a sort of a flexible probe molecule that can detect a wide variety. And then lastly, again, if you need to detect something pretty quickly, this is work actually from Gao in 2019. They're using this just for sensing odors in the air, but you can imagine G proteins are found all over the body. They're very fast. re-engineering these so that they're sensitive enough to detect things in vivo. So you notice this is very quick. You get a response within minutes, but it's not terribly sensitive. It's like micromolar, whereas most of the things we're going after nanomolar or sub, right? So different technologies that might enable fast response, generic response, et cetera.
I'm Adria Robinson. I am the agreements officer working with Paul on React. So I'll be the one working with any of you all who are selected on negotiating and getting your awards. So React, we are working under an MAI, Master Announcement Instructions. It's a little bit different from the traditional BAA that you're probably used to. So there are two documents, if you haven't seen them all already, that are really going to be your guide, and you need to use both of them when developing your responses. So the first is the MAI itself. The MAI is going to cover all of your high-level instructions. The MAI has instructions that are really going to apply to any module announcements that are released under it. React is the second module announcement that RSO has released. There will be others. So anytime you're responding to a module announcement, make sure you have that MAI document there in front of you. The module announcement is going to be your program-specific document. That's going to talk about, the technical details, in this case of React, and it's going to talk about your submission requirements, due dates, formatting, and that type of thing. Right. So within the MAI, if you've taken a look at it, you'll see we address module categories. And these module categories are designed to help you understand the basically the size and scope of what we're looking for in your proposal response. For React, we're looking at a gig module, which is between $25 and $50 million per award. So as you're putting together your proposals, that's the size of the award that we're looking at. And we're going to do stage submissions of your proposals. So you're not going to be submitting a full technical management cost price all at one time. The first stage will be just your technical and management. We are going to ask for a high-level basis of estimate, so we have an idea of, dollar-wise where your solution falls. And then in that stage, you'll also address any administrative national policy requirements. That portion of your proposal will be evaluated, and if selected, then we will ask you to submit stage 2, which will be your more detailed cost and price, any certifications, and at that point, we will provide you with a model agreement so that you can start reviewing terms and conditions and provide feedback and on those terms and conditions.
I expect your cheer to go up. You don't have to do it. You don't have to do a whole budget in the first proposal.
Trying to relieve the burden up front. So hopefully you all will like this. So there will be a bundle of attachments. That bundle of attachments is going to basically give you all of the formatting instructions that you need. So when we release the final, what's on sam.gov right now is a draft. When we release the final module announcement, there will be a bundle. And that bundle of attachments, make sure you read through everything, because you're going to have to submit most of what's in there, or at least submit to most of what's in that bundle of attachments. And you'll get another bundle if selected at stage 1. When we request your stage 2 submissions, you will receive templates for that stage as well. So for React, we are going to be awarding other transactions, which I know might be a little bit new to some people. So we're going to talk about other transactions here in more detail in a minute. As I stated, we're anticipating gig category awards, and all of the conforming proposals will have a scientific and technical review. They will be formally evaluated. Right now, we are expecting full proposal evaluations to be completed by March, and we'll start negotiating with selected performers in April. So since we are doing other transactions, we are outside of the FAR, Federal Acquisition Regulations. We're not doing any procurement contracts. We're not going to be awarding grants, no cooperative agreements. So all of those regulations that govern those types of awards that you all might be used to in the past, throw it out. We are not dealing with all of the restrictions that come with those sets of regulations. So other transactions. ARPA-H has the authority to award other transactions. You can see on the slide here when we have that authority. Other transactions are agreements. So the awards will be negotiated between ARPA-H and selected performers. So I mentioned we're going to provide you with a model agreement. We do expect you to provide us with questions, comments, red lines that will essentially start the negotiations on those terms. With the exception of laws that we are, everyone is required to adhere to. Think things like Civil Rights Act, right? Those are things that I can't negotiate. Outside of that, we have the flexibility with other transactions to negotiate essentially anything. So the idea here is to come up with an agreement that is mutually beneficial. And so if you have questions, if you have comments on the model agreement that we send you, please do not hold back. We encourage open discussion because that's how we're going to get to an agreement that's going to get you all what you need and get Paul what he's looking for on this program. So I mentioned that we're staying outside of those regulations that we're all used to. You can see here we list some of the regulations that don't apply to other transactions. We are going to be, a lot of you all are working in the commercial space. And so we are open to adopting some of those commercial practices because we find that they just work. And if that's going to get us a smooth execution of this program, then that's what we want to do. So we can negotiate payments, we can negotiate intellectual property, those types of things. It's a very streamlined award process. On our side, we'll have everyone involved from day one, and we hope that you do the same on your side so that once we start the negotiations, we can move through it in a really efficient manner and get to an award. The only downside to other transactions that we have seen is that it's just most people aren't familiar with them. So there is a bit of a learning curve, but we will walk you through it every step of the way and make sure you're comfortable with the outcome. So the process for REACT, we are requesting abstracts, but submission of an abstract is not mandatory. I would highly encourage you to submit abstracts because that's going to help inform whether or not you should spend the time and money putting together a full proposal. Abstracts for REACT should not exceed 6 pages, and that excludes the cover page and the ROM that we ask for at the end. There is a template for the abstracts in the module announcement, so make sure you don't leave out any of the sections of the template. And the abstracts are going to be submitted via ECPS, and there should be a link to ECPS on SAM for you. The full proposal will follow the stage submission process that I talked about a minute ago. Those will also be submitted via ECPS. And all of the page restrictions and other formatting requirements are going to be in the module announcement. So the evaluation of your proposals, there are 4 evaluation criteria that you'll see listed in the MAI. So your stage 1 submissions are going to be evaluated against criteria 1 through 3, and those are listed in the MAI in descending order of importance. Your stage 2 submission, as I stated before, will only be submitted if we request it, if you're selected at the stage 1, and that submission will be evaluated against the 4th eval criteria that's listed in the MAI. Right, so here's the eval criteria. I'm not going to talk through them in detail, but the first is overall scientific and technical merit. Second is capabilities and related experience. And again, these are listed in descending order of importance. The third is potential contribution and relevance to the mission. And then the 4th, which again only applies to stage 2 submissions, is the price and value analysis, cost realism, and reasonableness. Before I move on, I will say, I see a lot of people taking pictures of the slides. We are going to make the slides available after probably early next week. I'll have the slides posted on SAM, but we are also going to make available the recording of this Proposers Day. So feel free to take your pictures, but do want to let you know that you'll have access to the slides later. All right, so everything's going to be posted on sam.gov. Make sure you're monitoring that. We will be posting the final module announcement on sam.gov. If there are any amendments to either the MAI or the module announcement, those will also be posted on sam.gov. We're going to be answering a lot of questions for you today. We're going to be having a lot of hopefully really productive discussions, but make sure that if anything we say conflicts with what we post on sam.gov, what we post on sam.gov is what governs. So just keep that in mind. Non-conforming, I want to highlight this note here at the bottom of the slide. Non-conforming proposals will not be evaluated. So make sure you read through those instructions thoroughly. So right now, we have an tentative abstract due date of November 30th. That's going to really depend on when we get the final module announcement posted. We will not receive any abstracts under the draft announcement. So if you want to start working on them, or if you have started working on them, that's great. But please do not submit until we get the final announcement posted. And we have a tentative due date of January 15th for your stage 1 proposal submissions. Again, this is dependent on when the final announcement gets posted. We are going to be posting questions, the questions that have been submitted as well as the answers. As more questions come in, we will add to that posting. The MAI itself also has an FAQ document that's out there. So keep an eye on SAM for that to see if there are any MAI level questions that come in that we end up answering later. So when submitting your questions, make sure that you write the question as clearly as possible so that we aren't wondering what you mean and we get you an answer that you need. Make sure you don't include any proprietary information when submitting your question. We'll have to scrub those to make sure that we're not turning around and releasing your proprietary information, because remember, any questions that come in, we are going to answer publicly. So make sure your questions are sanitized. We might rephrase your question or consolidate questions if they're similar, but we're definitely going to answer everything that comes in. However, any questions submitted less than 10 business days in advance of the proposal submission deadline, we might not be able to answer in time. And similar to the comment I made about comments that we make throughout the day today, if there's any conflict between the FAQ and the actual MAI or module announcement, we're deferring to the MAI and module announcement. All right, so this outlines the review process. Abstracts, like I said, will be submitted via ECPS, and all of them will be reviewed after the abstract submission deadline. So the first step in that is compliance and responsiveness. Did you submit it on time? Did you follow all of the requirements in the module announcement? So if it is compliant, we will assign it to SME reviewers. Reviews will be conducted. Paul here will make recommendations, and the SRO, is that going to be you? Okay, so Jen will make final determinations, and then you will receive, an invitation or an encouragement to submit a proposal, or a notice that you're being discouraged. You can still submit a proposal if you're not encouraged to. That's obviously a business decision for you all to make, but we want to make sure you have that feedback so you kind of know where you stand before you do that. When proposals are submitted, so I missed this on the slide, we are not submitting via grants.gov. All proposals will be submitted via ECPS. And we will, again, do a compliance and responsiveness review for the proposals. They will be assigned to reviewers, evaluators. The proposals will be evaluated against the 3 top evaluation criteria. Again, Paul makes a recommendation. The SRO, Jen, will make a final determination, and then you will receive a notice of selection and a request to submit your stage 2 proposal so that we can begin negotiations. On this slide, we're going to just focus on the right-hand side. We're looking at the other transactions and what you're going to need to submit. So for volume 1, your technical and management, there's an administrative section, your detailed proposal information. And this slide says 30 pages max. I believe our max is actually 40. So we'll make the update here to the slide, but you have 40 pages. And then within the volume 2, if you are selected at stage 1 and asked to submit stage 2, these are the things that we're looking for. We're going to be requesting a spreadsheet. We'll provide a template for that, any supporting documentation to justify the numbers, the dollar values that you've included in that spreadsheet, your OCI disclosures and mitigation plans, if necessary, any rate agreement, any intellectual property representations, and then also that's where we'll be asking for your feedback on the model agreement. I believe this is my last slide, so happy to take any questions if you have any at this time. Subject matter experts. Mhm. Yeah. There's no cost share requirement. You are free to propose cost share, and that's something we can negotiate, but there's no requirement for cost share with the OTs. Paul, I'll turn that over to you and your team.
We're going to try to get everything back to you within a week, so they have plenty of time.
Sorry, the question was, what's the timeline for feedback on abstracts? And Paul's answer was they're shooting for a week.
Very curious what sort of feedback that we've provided with response to the abstracts. Response thoughts or
So yeah, so the question is, what sort of feedback is going to be provided? I mean, the baseline is, whether or not we think you have a reasonable chance, right? Pulling together a full proposal takes a lot of time, and so trying to make a judgment call on if you think you're actually going to get there. Some of it is an emphasis, right? You might have brought up something that is super clear to you, and we're like, you really need to explain this a little bit better, right? It sounds interesting. You're going to have to explain this a little bit within the proposal. Sometimes you're out of bounds, and you have to, we're going to provide some guidance, right? So we have to walk a fine line. We can't put words in your mouth. We can't, tell you what to put down on paper, but we can ask you to explain a little bit more. To refine here, then sort of an emphasis. Make sense? Okay.
You mentioned this will be a gig level program. Is that the funding range for that? Is that for all of the awards or for
For the gig level funding, the dollar amounts is per award, not for the entire program. Any other questions? Yeah.
the gig award?
Yeah, it's the award amount for the entire award. The entire all three, is it three phases, right, Paul? Yeah. Yeah.
Something about funding or funding.
Yeah, this goes back to something Jen Roberts said earlier. If you are proposing a solution that's already being funded elsewhere, it's not really something that's within the ARPA-H space to fund. Do I have that kind of? Okay.
Effectively, we don't want the same proposal funded by.
Right. Yeah, we're not going to have multiple government agencies funding the same solutions. All right. I'll be around all day, so if the other questions come up, feel free to just grab me and ask. Oh, yeah.
What drives the January 15th date? You said that could change?
Right. When the module announcement is posted, then we're looking at making sure that people have enough time to put together a full proposal. So if we don't get the module announcement posted when we want to, then that will shift that proposal due date a little bit.
Trying to do 60 days or 30 days or 45 days?
We were looking at roughly 45 days. So we're going to try to stick to that. Yep.
Okay, so it is my distinct honor to introduce Lieutenant Governor Diane Primavera. We're honored for you to be here. We first heard about all the cool work that you and your office were doing at Cancer X with sorry, with Stephanie Pugliese. That was at a meeting a couple months ago at the White House, which is pretty fancy, but not as fancy as the Brown Palace. This is an amazing video. But it was at that time we heard many of the things going on in the office. They're doing great work and connecting hospitals and rural settings with clinical settings in large cities, which is something near and dear to ARPA-H. Also, from her bio, you learn for triumphs over cancer, her leadership in the Susan B. Komen, and from that taking a strong personal desire to help everybody fight cancer, right? Empowering every last person, which again is near and dear to the heart for ARPA-H. And so we read all these, we're like, we really got to talk to this woman. So we're really honored for you to come here and be with us. And I know you have a couple words you wanted to say. Thank you.
I'm never as tall as the speaker before me. So anyway, good morning, everybody, and thank you for inviting me to join you today. I'm amazed at the amazing minds that are in this room. As a Lieutenant Governor of the great state of Colorado, I oversee a diverse portfolio of programs that support and advance the Polis Primavera administration's health priorities. I serve as the director of the Office of Saving People Money on Healthcare. And if you can come up with an acronym, I know you're all brilliant. Maybe you can help me on that. And I also lead the Office of eHealth Innovation, and we call that OHI. I've dedicated my career to championing high-quality, affordable healthcare that all can access, and I'm proud to help lead the Polis Primavera administration's vision of a Colorado for all. This means that no matter your hometown, your income, your race, your gender, your ethnicity, your age, your disability, your veteran status, or sexual orientation, you can thrive in our state, which includes reliable access to affordable, high-quality health care. I know that the cutting-edge work that you all are doing is an essential part of achieving this vision. ARPA-H combines the latest in scientific research with the power of innovation, aiming to not only improve people's lives and health, but to save money on health. Myself, as someone who has received the challenging news of a cancer diagnosis on 4 separate occasions, I know how rocky the road to wellness can be. And the fears of missing milestone moments for my daughters, balancing a job with treatment, and dealing with the stress of bills from the high-cost medical care, I needed to beat cancer. And I know navigating today's world with a chronic condition is a huge burden to bear. And when I was diagnosed, I didn't have anything that would help my T cells. So I used visualization to try and beef up those T cells to fight my cancer cells. And as a cancer patient who's been lucky and hasn't had any cancer scares in many years, I'm continually reminded that each day I get to be here with my daughters and my grandkids and serving the state I love is an incredible blessing. Time is one of the greatest gifts that we have, and with the development of new health technologies, not only can health conditions be more easily managed and treated, but people will likely be able to spend less time at the hospital and more time with loved ones in the comfort of their home. As a survivor, I feel that it's my calling to speak out for the millions of people in the U.S. living with cancer and other chronic conditions so they can have access to science-driven, high-quality care that they need to lead the best lives that they can. Colorado is home to research and innovation across all fields, and we know this is the type of work we need to move healthcare forward. We have a strong bioscience industry, and it's an area we're continuing to partner with here at the state. Earlier this year, Governor Polis reauthorized the state's Advanced Industries Accelerator Grant Program, extending the program for a second decade. This program has been critical to launch innovation in Colorado across many innovative industries. In the program's first decade, the program created over 5,000 jobs and retained over 5,200 jobs. In May 2023, the Advanced Industries Accelerator Program awarded $3.5 million in grants to Colorado bioscience research institutions and companies alone. Innovation and research like the REACT program not only solves important medical challenges, but also plays a crucial role in creating a thriving economy in Colorado. Together, we can support new technologies that make it easier for people to get the right treatment that they need and when they need it. This means better outcomes for patients living with acute and lifelong diseases, and for those who routinely have to travel far from home to receive care. The work you all are leading through ARPA-H and the REACT program is incredibly valuable, and I'm glad to join you here in Colorado in support of it. So thank you for your time, for your passion, and have a great day.
All right, thank you so much. I think what we're going to do is we're going to take a brief break. For those who've never worked with an ARPA before, one of the great things is you do your job of delivering the technology, and the program managers and the team are out there trying to change the world to make sure it's going to thrive. One of the amazing things that ARPA-H has done is set up the patio office that Jenica is going to tell you about, a huge catalog of capabilities to make sure your technology thrives out loud.
All right, great. I'm so excited to be here, everyone. Thanks for sticking around. I know I'm actually the one that's holding you back from lunch. So, oh, there's 2 more people after me. I actually will talk about them. So, like Paul said, I work in the transition office. I am the director of the Technology Transfer and Transition Division. And I just wanted to tell everybody why our office is so unique. So we talked a little bit about translation early in the morning, and how I define that is really moving from bench to animal models to humans. How we define transition is taking that MVP, or that minimal viable product, and getting it out to the hands of all people. And why that's so important for us is because it's really going to help us meet our mission to accelerate better health outcomes for everyone. So we are this unique technical office in ARPA-H that is able to help facilitate that with our program managers, as well as our performer teams too. We're very unique in the sense of an ARPA because we have a very complex transition partner. It is all Americans. That's hard because it's 300 plus million people, but it's also hard because we have to jump through a lot of hoops to help accelerate better health outcomes in health. So that's regulatory and reimbursement, just as one example. So really the mission of our, I don't know if I can move this thing, but really the mission of our office is to be able to enhance that probability that the technologies you are all developing will be able to get out to the hands of all people. So we are there to identify barriers, and we're also there to provide transition support and commercialization services to our program managers, but also to our performer teams. And we know that we're not going to fund all of our performers in perpetuity. We are not like NIH. We don't keep a program around for many, many years. As Paul talked about, 5 years, we're ready to transition that technology right after that. So we are hoping that our services will support Paul and will support the performers on this program to be able to make sure we can maybe attract private investment for you or get you to translate those breakthroughs. So how Patio is set up, we model the IDEO framework. So we look at a human-centered design approach, and we use these 3 different ideas that we think is going to enhance innovation. And that's feasibility, so focusing on the technology. That's what Paul is doing here. He is our technology expert, and you here as well can help identify what those technologies are. Where the secret sauce is for Patio is really in the desirability and the viability. So desirability is really creating technologies that we know people want to enthusiastically adopt. And then viability is really, we want to make sure we're developing technologies that the market wants to enthusiastically adopt. So we've created a lot of ways to be able to make that happen. So you're probably asking yourself, well, where might the solution that I might develop for Paul's program might go after ARPA-H? And I really want to encourage you while you're developing your proposals to really think about that. Who do you think your transition partner would be? Do you think you're going to take it to a small startup, maybe out of an academic group? Do you think that you might integrate into a healthcare system or go to a nonprofit or another government organization? Again, we are here to support that, but we want you to think about that while you're developing your proposals and think about what are the barriers that you're going to go through to actually break through the barriers and make that solution successful. So so everyone thinks about transition as at the end. You create this cool technology and you're going to transition it at the end. That's not how we think of it. We think of it as we are going to be there at the beginning with our program managers while they're developing their program to be able to make sure we're integrating in transition ideas and understanding what those barriers are from the beginning. So we support transition through the entire pipeline of the program. So right now we're in between solicitation development and early program performance, and you'll actually get to meet 2 people right after me that have helped support Paul in developing his program. So within my division, we have 3 divisions in Patio. My division is the Tech Transfer and Transition Division. We've created something called the Experts in Residence program. So think of that as really private sector expertise that really can increase that viability, as I talked about earlier, through expert advisors and support. And we're hoping that's going to be a highly specialized bespoke service for our program managers and potentially for our performer teams to make sure that they are thinking about the transition and commercialization. So we can embed the experts with our teams, with our internal to ARPA-H, as well as with our performer teams. So if you're chosen to be an awardee with Paul's team, you might be able to get this service. We also have expertise in regulatory and reimbursement support. So we know that we want to make sure that these technologies are affordable and accessible, so we can provide that expertise to help with that. And we also want to make sure that this is accessible to all Americans, including marginalized communities, et cetera. So we want to make sure that we are pulling in equity-focused experts as well. You'll get to meet these 2 expert in residence that supported Paul. One is Imran. I don't know if you're here. You can raise your hand. He's way back there. He's going to talk soon. Comes from Digitalis Ventures. And then we have Natalie that's sitting right here. So they are actually funded through one of our public-private partnerships from Digitalis Commons. And they're going to talk a little bit about how they're supporting Paul. Another division that we have is called HEAT. It's our Health Ecosystem Engagement Team. And this is really helping with desirability. So we want to make sure that we have access to resources and expertise of our health ecosystem through a network on demand. So we've been reaching out to people across the entire nation. We actually sat with people yesterday in Colorado, from universities all the way to tech transfer groups, to bring people into our network, because we want to make sure we're connecting with the right people at the right time to be able to help our program managers in our programs. And the last division is our ARPA-Net for Health. We just launched this at the end of September. Really excited, a lot of buzz about it across the nation. But really, our goal is really to meet people where they are. And we think the ARPA-Net-H will help do that. So we have a couple hubs across across the nation that I'm going to talk about in the next slide. But we want to make sure that we're developing solutions that are going to be desirable and are going to be viable for the nation. So what Congress actually mandated that we had 3 sites across the country. So we took this as an opportunity to actually level up and connect the entire nation. So we have one operations and stakeholder hub that's in Washington, D.C., but we actually solicited out to see who also wanted to be our other hubs across the country. So we have one that's our customer experience hub. So again, getting to that desirability, looking at a human-centered design approach to support our technologies. That's located in Dallas, and it's supported by an other transaction or contract management firm called ATI. And then we have our Investor Catalyst Hub, and that's in the greater Boston area. And the Investor Catalyst Hub is going to help us with that viability. So really making sure that we're creating technologies that can be viable in the market. And that is run by VentureWell. People might know VentureWell from NIH. They ran the RADx initiative. So how we're making it an ARPA-Net for health is we want to make sure we're connecting to the entire nation. So not only do we have these hubs, but the two hubs I talked about have spokes. Actually, one spoke just talked about today in their little lightning talk. So that was awesome to see. But you're probably asking yourself, well, what is a spoke? Well, first, a hub is an actual physical space. They are really our nerve center for the network. They help convene these networks and these spokes. Our spokes are the members of our network. They are what we call the muscle of the network that actually make these things happen. They are going to bring those experience and expertise to help our performer teams that you might be at some point to be able to get that expertise and that support to transition their technologies. A spoke could be somebody from industry, a hospital, an academic institution. Anyone in this room, you could be a spoke. If you're interested in becoming a spoke, you actually can go to our website and sign up. If you think your expertise lies in the Customer Experience Hub or the Investor Catalyst Hub, really easy application. Go to our website, sign up. It'll be awesome for you to be part of our network. So I just have 2 more slides. I'm going to go a little bit deeper in what the Customer Experience Hub could do for a performer team as an example. So something we could do is do immersive experiences. You're developing this cool technology. Have you ever been to a rural hospital to see how they implement it? Will it be implemented? What if we could go and do that and make sure that maybe we're not designing that technology properly? So we could go to that rural hospital to see if we can design it a little bit better to be implemented in every place in America. Another example is novel clinical trials. So Jen, I think, or Paul mentioned earlier, ACTOR, Advancing Clinical Trials Readiness. That's actually a program that we're launching through the Customer Experience Hub to help make clinical trials more accessible to all Americans. And then lastly, our Investor Catalyst Hub. It's really around overcoming common business problems. So something you could think about is, what if we could do education and training with the performer teams to support that commercialization? Or what if we can get you that regulatory and reimbursement support that sits with your team to make sure that you're making something that is more affordable and more accessible to all Americans? So that's just a few examples. So that's all I have, but I'm going to hand it now over to our experts in residence who've been part of Paul's team. I'm going to first, it looks like Natalie is first, and then Imran, you'll be next. So Natalie, if you want to come up and talk a little bit about what you're doing. Thank you.
Great, thank you. I thought I was going to be the thing standing between you and lunch, but that's not the case anymore. Could you advance, or can I advance myself? Oh, there we go. Okay. So I'm here to share some lessons learned from my entrepreneurial journey in developing a long-term implantable sensor to go in the body. And that was with Perfusa, the company that I founded. I'm now at a venture capital firm, Material Impact, and I'm also supporting the REACT program and ARPA-H through Digitalis Commons. And so my background to bring to bear to you is I had studied biofouling and foreign body response in my PhD in the 90s at Duke, then went to McKinsey for a couple years and then started my own consulting firm for medical device, looking at quality, regulatory, technical strategy, and so on. But the really relevant experience to bring to bear is with founding of Perfusa to monitor biochemistries in the body real time and longitudinally with permanent implants. So we pioneered tissue integrating sensors, this idea of soft, flexible, porous sensors in the body that would integrate was our technology, or I should say is our technology. And I took the idea from concept all the way through commercialization, through to regulatory approvals, and the technology is still, the company is still going in the hands of capable business folks. We built up a 100-person team, including contractors, and raised $90 million, much of which many thanks to DARPA and NIH for getting us off the ground. And I was PI on those awards and a DARPA performer. And now I'm in venture capital. And the image that you see here is actually a histology slide that contains our sensor. And it's not the red dot that you see there, that's actually a hair follicle. So our sensors are so tissue integrating that sometimes it's hard to distinguish them from the actual soft tissue structures. And our sensor is right in the middle, right there. So that's what I worked on. And so my experiences and many mistakes that I made along the way to take the idea from concept all the way to clinic, I'll be happy to share what I can here. I'm going to go one by one with each of these, and we'll review the list at the end. So tip number one, don't let the body be a black box. So many sensor developers, cell developers do all kinds of things in the lab and characterize their cells and their sensors, and then they put it in the body. They don't really know what's going on in the body around the sensor or around the cells. So don't let the body be a black box. Embed as much sensors as you can, external sensors, imaging, wearable sensors, know what's going on, characterize that physiology as well as the local environment. And if you don't have many multiple data streams coming out from your implants, regardless of whether it's a living pharmacy or a Sentinel, you're not getting all the power you can with the investment to develop what you're developing. Really invest in ways to monitor what's going on continuously. Second tip, don't underestimate the foreign body response. It will kill your device. It will kill your cells. It will kill your sensors. And this is an image from the front cover of a biomaterials journal or magazine. And it shows a hip implant that is pitted with divots where macrophages resided, and they were activated, nasty macrophages that when they don't like the environment that they're seeing, they can actually drop down to a pH of 2 at the biomaterial interface, and that can do real damage. So now, not all macrophages do that. You have to keep your macrophages and all the other immune cell types happy, but that just goes to show you how important it is to pay attention from the beginning how you're going to control the immune response to your implanted device. Okay, tip number 3. A pig is not a pig is not a pig. So it's widely known that the accepted model for subcutaneous implants and skin implants, the pig is the preferred model because it most commonly mirrors the immune response in humans. But I learned this from Becton Dickinson, who is a powerhouse receptacle of a lot of preclinical knowledge, that even different strains of pigs react differently. So your Yucatan mini pig versus your Sinclair mini pig versus there's other breeds and brands, and there's your general cheap farm pig, very different from some of the other strains in how the immune response develops over time to an implanted material. So pay attention to strain as well. This information isn't widely available. Nobody really publishes on that widely, but it is very important to consider. And rats, everybody goes to rats, not really representative of what happens in humans at all. It's very fibrous tissue compared to humans. I actually have a paper published from my grad school days on rat, pig, human comparison. Okay, moving on. This is more on the business side. Choose your vendors, your suppliers, your developers, choose them wisely and manage them. Just from personal expertise, anybody who has a pathology lab will say, oh yeah, I can do those stains, I can do that. But if they don't look at biomaterial surface implants on a regular basis and have a system to do that and metrics, you're going to take 6 months or a year to develop them. And that's true for pig models or any other animal models or CRO models. So choose people wisely for their expertise. Have a point person internally to manage them on your team who lives and breathes the experience with those vendors or suppliers or partners. And I like hire slow, carefully, but fire fast. If something isn't working, really manage to what you need. Okay, number 5, validate sensors relentlessly, and cells too, but I can speak from the sensor side. These are images from a single pig, one pig, 2 different sensors And you can believe those sensors had vastly different performance and readings because the one has a significant number of immune cells around it, the other very different. So if you have fibrosis developing, or if you get a lot of microvasculature very close, or that the tissue development changes around the sensor is what your sensor is going to be measuring. So you have to have good controls, know what you're measuring, and think longitudinally. How are you going to have controls longitudinally? Super important, blood is not a great control. It does not mirror interstitial fluid, what's happening in the tissue, except for the few beautiful cases like glucose when everything is right. But I've worked on lactate sensors, pyruvate, glycerol, urea, oxygen, hugely different kinetics, hugely different concentrations. So blood doesn't equal tissue. In vitro does not equal in vivo. In vitro, you develop your sensors in a fluid, maybe in a gel, but the mass transport is very different than in tissues, right? The diffusion rates of the different nutrients and waste products, very different in vitro and in vivo. Even though you should try to model it, it's difficult. So get to animals as fast as you can. Okay, electrodes biofoul, fluorophores photobleach, and there's just a lack of molecular when there's a lack of microvascular proximity or fibrosis, you just have limited transport properties. So everybody can like produce that single beautiful image and sensor for a paper. Anybody can publish, but can you do it reproducibly? That's super important. Show all your data, be honest about it. It's super helpful to really solve the problems that are going to get you to the next level. And that goes very much to my next point, tip number 6, de-risk, 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 right? If you want more funding, you have to de-risk and understand both the ARPA-H mindset, all your technical hurdles, you got to, meet those hurdles, but then think about it from an investor standpoint, what kinds of things need to be de-risked to make a VC or a strategic partner want to invest in you 3 years, 5 years, 6 years out. So think about constantly what you're de-risking. And then tip number 7, this is very much more on the business side, but it's for everybody. Really invest in your messaging. What is your story and how do you tell your story? It will serve you well, whether you're a scientist or a CEO or a professor. Tell the same story consistently. Or if you change it, have a good rationale why that's communicated and tell that story at the right level, at the right time to the right people, and it will really serve you well. So in conclusion, my 7 tips, number 1, don't let the body be a black box. Number 2, don't underestimate the foreign body response. And number 3, a pig is not a pig is not a pig. Number 4, choose your providers, vendors, experts wisely and manage them. Really, big bold, manage them. You're going to get a lot of money. Use it really wisely. Validate relentlessly, both your cells, what they're doing, and your sensors. De-risk as much as you can, everything you can, and then invest in messaging. And my email is there. I'm happy to talk here or talk later and share many of the mistakes that I made in hopes that you don't make them again. It's going to be a really exciting road. Good luck.
All right. Hey, everyone. So I am going to speak to you about, and actually the last 2 talks have been perfect in sort of the setup for this, but all these great ideas, and I especially want to sort of speak to folks from an academic lens as well, but all these great talks that we're having and ideas that are starting to generate, right? These are going to start creating innovation. And that innovation is ultimately, if it doesn't get to patients, like what is it that we've really accomplished, right? Unfortunately, that journey requires a travel through industry, right? So somewhere between academia and to patients, there's going to be a journey through industry. And that's the lens I sort of want to share with you as to what that looks like. So who am I? Just really quickly, I've spent about 20 plus years working in life science on sort of how do you access innovation in different ways. I spent a lot of time in pharma and business development. And in the last 10 years, created a venture fund called Actual Potential, which was really trying to look at the intersection between biology and engineering specifically, and how could you treat disease without the use of drugs. That was the idea. We focused originally on nerves, but really today, the idea of like, can you treat any disease as long as you're not using a drug? And what that has done essentially is over the course of the last 10 years is we've built out a portfolio, and much of that portfolio was in seed and Series A companies, which were coming from academia. So every year we take a look at about 2,000 opportunities, and we invest in 2 of those, right? That's about the rate that we sort of operate in. And many of those ideas that we don't invest in, I want to share a little bit about why that is, right? So, but before I do that, I do want to highlight that to me, like I think what Paul and frankly the patio concept that's built on top of it, I think it's really, really fantastic. But from someone sitting on the other end of this lens, from the venture capital side, like why do I care about this, right? And to me, when you still today, when you think about medicines and all these wonderful medicines that are out there that people aren't actually taking, this solves a very big part of that problem, right? And so from my lens, and I would encourage this for all of you to always keep thinking about what is that unmet need, because that is what's always driving us. What is the unmet need in whatever you're doing? And is that a big enough problem that you're solving? So I think that really, the REACT program really addresses that. Another development, and that what we've seen in the last 10 years with bioelectronics, and especially during the COVID years as well, sort of remote patient monitoring and the potential for what that means has been huge. We've seen this now sort of consistently, irrespective of disease area, that when you put the patient in a place where they have greater agency in that decision of what they need to do with their therapy, the results start changing, right? And so we've seen this across the board. You may empower the patient and you start seeing better response rates as well. So I think the idea is, as we start thinking about that here as well, very, very exciting. I already love the invitation that many people are putting out there, like for us as well, trying to bring biology and technology, engineering, and various facets within bioelectronics is really, like you set out to do one thing and you actually realize, of course, that you're uncovering sort of things that you hadn't even sort of set out to do, right? And so to me, when you think about personalized medicine, remote patient monitoring, a lot of the ideas of where sort of we see healthcare going, I think the REACT program really sort of checks a lot of those boxes. So that's the really good stuff. That's why I'm excited, waiting to see what you guys come up with as well. But what are the things that you need to be thinking about as you make that transition from academia to industry, right? And so I do have sort of 12 things that I was thinking about. I'm going to focus on 6 of those because I really do want to drive this sort of point home. But when we're pitched with an early stage idea of something, hey, would you guys be interested in doing this? Often the problem is that give us something that we can rely on, something that you can have a little bit of evidence, right? We're not expecting it to be everything, but a little bit of evidence, that you have a proof of concept here that works. And I think of all the things that I'm going to say, if I could say this, and I like the way Natalie was repeating this as well, but all the things I could say, I would just say commercial viability assessment over and over again. And I know when you're sitting in academia, someone's insisting that you think about commercial viability, it seems a little ridiculous. And I part of me wants you to ignore what I'm saying when I say that, because you should pursue the science for the science. But if we are not thinking about who the patient is at the end of this, then we are doing things that we are not sort of making sure are guided. And so of those 2,000 opportunities that I turned down, many of them are the classic, it's a great hammer looking for a nail, right? And it's like, and I don't want, I'm not interested in that nail. So like really, as you're continue to sort of think about this idea of ultimately what would be the patient population that I'm thinking about, what are the other options for these patients, and why are the things that we're doing are interesting. So commercial viability assessment is so important, right? Scalability, I heard that today as well, but scalability, especially for technology as complicated as what we're talking about here as well, does need to be scalable so that it's practical from that perspective. Intellectual property, and again, I'm sort of directing this from an academic lens, but thinking about intellectual property for us from the earliest parts are always something that are important. I'm going to touch on regulatory separately as well, because I think it's a real sort of challenge, particularly here. And then clinical validation is perhaps the last thing I'll touch on here. Clinical validation, and I know sometimes ideas are very early, but we say that, look, if you can even give us a very rudimentary sort of indication that in a patient population or in a biomarker that we could use, something that can indicate that if we were to do this thing, that you're seeing a clinical effect that matters, is a very important question. And the quicker we can answer that question, the more likely it is that we're going to step in. So like I said, there's there's other things as well, which I can talk to, but I really did want to stress those first six. And jump over and come back to that slide, but to talk about the regulatory challenges and the two things that are in the last 10 years that have made me the oldest and perhaps most tired is the challenges on the regulatory side and the reimbursement side, right? We have so many wonderful technologies out there, so many wonderful therapies that are not getting paid for and are not finding a way through the regulatory hurdles. And why is that, right? And it's because, it's often because, and it'll be the case here for React as well, that these are very, very novel technologies. Is this a drug, right? Is this a device? That's a question. Who within an FDA body would then regulate that? As you're sort of moving through this, think about the information, especially with remote patient monitoring and the ability to communicate. Think about the information these devices may be collecting and are then transmitting, right? So how are we protecting that? Who is ensuring that that protection is there? Long-term safety, these devices, conceivably, pharmacy on board, sort of how long do you need to show safety for, right? And a lot of sort of speaks to this fast evolving technology space, which is that often the technology is moving way faster than where the regulators are. And we have had situations, and it's been, to share we've had situations where we have taken things to the regulators and they have said, we can't approve this for guidelines that we haven't issued yet. We will issue those some date in the future. But when we issue them, we don't think you're complying with those guidelines, right? And it's like, but like, how would we know what those guidelines are, right? But they're like, well, wait for 2 years. And if you're a venture-backed company, it's a very impractical thing to ask for us to do, to say, well, let's wait for 2 years. So the regulatory challenges here will be just given the, I'd say the complexity, but the beauty of sort of what's being put together is going to be high. And that's just something that we need to be thinking about upfront. But let's assume that we get through the regulatory part. You're also then going to have to commercialize this, right? And ultimately the 300 million plus Americans that are going to be the users, the customers, the patients of these therapies are going to have the same questions, right? Data security, privacy, where is this data going? Who has access to it? When we talk about user acceptance, I will say that often means the physician more than it means the patient, because physicians are trained in medical school with the things that they know. And even when they know that things don't work, they know those things. And so they are willing to prescribe something that doesn't work sometimes before they're willing to be convinced of something new. And it's just, that is part of the process. And the cost and reimbursement, and I'm not here to sort of have a big dialogue on reimbursement, but when you think about how reimbursement companies think about paying actual claims, they call it medical loss, right? So when we actually submit a claim for healthcare delivered to us, that's a medical loss to an insurance company. So from that perspective, keep in mind that they are always looking to keep their costs down. And so we have to be mindful of that as well. So how do you build a health economic argument around this particular therapy? And how can you really sort of make that case? And so there's going to be a lot of education required. There's going to be a lot of public perception and ethical considerations that need to be thought through about all that as well. But I say all that, I know those are huge, those are discouraging ideas as well, but like the hugeness of the idea that is being proposed here and what this could actually have, and there's so many elements of it that are starting to come together, I think could be absolutely wonderful. So the only sort of guidance and sort of thought that I would say is that even in the depth of academic research, and as you're thinking about that, every so often do step up and sort of look in the distance as this something that is being administered to a patient in the future. Who is that patient? What are they doing? What are their options? And ask those questions of yourself to make sure that you're sort of still going down a path that makes a lot of sense. So I too will be around, happy to have a conversation with anyone, but thank you so much.
We do have one final talk from the Advanced Regenerative Manufacturing Institute. Again, these are the types of capabilities that we like to bring to bear in these programs. He's going to talk about some of the advances of their capabilities. So one of the things I'm thinking performers in this program would be working with Army to help validate the technologies, think of ways to advance the commercialization. And I promise Tom is the person that we treat you at lunch, but he's going to be great.
Thank you, Paul. And I don't want to get into an argument about who has more power in keeping everyone from lunch. Paul's given me 10 minutes to speak. He doesn't know that I can easily go on for several hours about this topic, but I will try and keep to my time. I am Tom Bolenbach. I'm representing the Advanced Regenerative Manufacturing Institute, ARMI, in Manchester, New Hampshire. We're a nonprofit organization. We're member-driven. We have 200 member organizations that we draw on to help us solve problems in scalable, consistent, and cost-effective manufacturing of cells and tissues, and eventually organs. We also sit in the space between academic funded research and industry, and try and fill that valley of death, as the DoD calls it, to try and bring technologies through to understand things like how do I scale up a living product? What is it that I measure about that product to ensure that it's going to be functional in the patient? In other words, how do I QC something so that I know it's going to be efficacious? And then building systems to enable manufacturing of these complicated products. We've heard a lot about reimbursement. We've heard a lot about the regulatory pathway. And I think where we sit is to enable you to get through those hurdles in the most efficient way possible. The thing that links regulation to your manufacturing and things like scale and whether this is ready to go into a patient is a quality system, for example. And so those are all the types of offerings that we have. And I'm going to try and build this as to make it as specific as possible to the REACT program. And so for a long time, and other people have talked about this, one of the things that's been a barrier to using cells as a medicine has been manufacturing. So as early as 1976, Eugene Bell at MIT published on the production of skin. And think about that. And in between 2013 and 2014 alone, there was a $3 billion investment by the federal government into the development of basic research into cell-based therapies and tissue engineered medical products. And that's and the challenge there really has been the manufacturing barrier and the scale-up barrier. And part of the challenge has been exactly what you see in front of you. So I wanted to provide a contrast between research and development in these products and manufacturing, what it looks like today. And it was completely accidental that I found these essentially mirror images of technicians in a laboratory that one doing R&D and the other doing manufacturing that looked almost identical. And the challenge here really isn't the use of the clean room or the fact that the manufacturing tech is using a biosafety cabinet and doing everything manually, although we want to address that as well. The other part is the question of yield. So the technicians using a T150 flask or a T225, and let's just argue for the moment that yield of cells after about 7 days of culture from that flask is about 10 million cells. So, you know, that's fine if your product is, you can provide enough doses to patients with a flask or a few flasks of 10 million cells. And if you're talking about something like a third line CAR T therapy, today with a few thousand patients treated to date over the last couple of years, maybe that kind of approach to manufacturing is okay. But one of the projects that we're working on currently for the Department of Defense is the production of red blood cells. There's 300,000 administrations of packed red blood cells in the US annually, and each unit of packed red blood cells has roughly 2 to 3 trillion cells in it. That's more than the output of the average cell-based therapy company over a couple of years, right? So what we need to think about is innovative ways to be able to scale these things up. And this is what I want you to think about as you're developing your products. How am I going to generate enough cells? What do I need to start thinking about to be able to generate enough cells to treat all the patients I need to treat? Because as someone else pointed out, it can get reimbursed, it can get through all the clinical trials, but if you can't make enough of it to treat patients at a reasonable cost, it really doesn't matter. It won't be for everyone. So our approach from a technical standpoint has been to adapt processes to scalable manufacturing equipment. I'll talk about that. We integrate that equipment through supervisory automation to essentially build manufacturing systems that can produce tissues from end to end. We close process to mitigate the risk of contamination that's typically associated with something that you would do in a flask And that really changes the manufacturing environment. No longer will you need a clean room to be able to produce these things at scale. It'll be done in a much lower grade environment. And someday, my hope is that we can manufacture cell-based products in a space like this. One thing I'll also emphasize is objective in-process and final product quality control. We think we're really smart, and we can predict what our critical quality attributes and our quality control specifications for these products need to be. That's really difficult to do based on a hypothesis, and that's failed time and again. And you only have to look at Fierce Biotech to see how many companies have been stalled in their regulatory path based on a lack of a good potency assay that the FDA and other regulatory agencies will be able to buy into. And then we also, and I won't talk about this too much today, only to emphasize that we work on a very broad spectrum of enabling technologies. We do fill technology gaps through technology development projects. We have about 40 of those right now, and those are enabling across the entire manufacturing spectrum. This just outlines the generalized manufacturing steps for any cell, tissue, or organ manufacturing, starting with cell harvest, either from a biopsy or a blood sample, expanding cells, combining them in some instances with a scaffold material, allowing that cells plus scaffold to turn into a tissue over a period of time, preservation and packaging of the final product, figuring out how do I get it from the NMI assembly line to the site of administration, and then how do I transport it there. This is our concept for scalable, modular, automated, and closed manufacturing systems. You can see the general steps of the manufacturing process highlighted at the top. All I'm going to say is that we use a modular approach using essentially what the biopharmaceutical industry can give us in terms of manufacturing systems, and then fill gaps where necessary so that we make sure that we're thinking about how to produce cells as a medicine, not necessarily producing cells that make medicine. Although in this case, Paul's program is definitely blurring those lines between the two. On the right side is where the first two sets of modules are very general. We can grow any adherent or suspension cell type. It doesn't really matter. We borrow and combine systems there. It's on the back end where we begin to give the tissue or the device, the implantable device specificity, where we begin to do custom engineering on these systems to be able to enable that scaled up and closed manufacturing. And I just want to highlight this robot arm. That's one of our member organizations, Advanced Solutions Life Sciences. And the only reason I highlight that is to give you an example of where we took a manual process from a lab at MIT that was using a synthetic biology approach to produce livers, miniaturized liver organoids from induced pluripotent stem cells. We took their process and we completely automated that process to culture the stem cells, dispense the stem cells into 96-volt plate format, and then be able to pick the resulting organoids with the arm into a microfluidic chip and place those in. So think of a conveyor belt producing microfluidic chips that have living organoids plus vascular cells in them as well. So very much thinking about the challenges that you're going to face in this. And this is an example of our prototype line. This one starts on the left side with a cryo vessel where the technician loads frozen vial of mesenchymal stem cells. The process runs through all the modules autonomously for 45 days. And on the back end, it produces bone ligament bone hybrids for implantation as an ACL repair. We do technical project calls periodically. And as I said, we have roughly 40 projects underway, some of which are maturing into enabling products. These are some examples. Closed centralized culture control systems for autologous production, in this case bone, but applicable across multiple tissues. We do a lot of screening platform development. In particular, our interest is in converting culture media to, as the biotech industry has done, to chemically defined media. And that's not easy to do while maintaining function and phenotype of the cells. So building a high throughput methods to be able to do that, similar to that, developing chemically defined preservation media. I hesitate to say cryopreservation because we're looking across a number of temperature regimes. And for the first time, for example, we partnered with one of our member organizations to be able to preserve pancreatic islets at sub-zero temperature as clusters, and then bring them back for implantation without any compromise in their ability to secrete glucose or to keep integrated clusters. One of probably the biggest focus areas for us is on non-destructive measurements of sensors. Again, this will resonate with you. In our case, we use those sensors to monitor manufacturing processes, and we have a variety of non-destructive ways to do that. Some of the things that we look at are specific, metabolites, proteins, and lipids in culture, VOCs to measure the presence of contamination in real time, to cut down on the amount of QC time that we need at the end of the process. With all that sensor technology, the question is, what do we measure? And in some instances, you're going to say, well, if I'm producing a therapeutic protein from this device, then I need to measure that therapeutic protein. But is that enough? So we talk about stem cell derived pancreatic islets. We work on that. We built automated systems to produce it. And it's obvious there that islets need to secrete insulin when stimulated with glucose. However, is that sufficient to enable engraftment of the islets? Will they continue to secrete insulin after implantation? For how long? Can I predict that in my QC ahead of time, which really is the function of QC? So we have built a core facility that we'll use to help validate some of the technologies here to be able to answer those questions. And instead of using hypothesis-driven approaches to understand what is it that I should QC about this product and try and predict that, I can predict efficacy in the patient. We ask not what should I measure this, should I measure this, should I measure this? We just measure the full multi-omic profile of the cell product, and then draw correlations between those data sets and either preclinical or clinical data, and use the modern and emerging tools of advanced computation to figure out what the subset of those data points are that are going to be predictive of efficacy. And once we understand those types of things, then we can go back through the process at each step of the manufacturing process and ask questions about whether or not specific markers along the way correlate with final product quality. And then once we know what those are at each step, then we can begin to ask questions about material attributes and process parameters that might affect each of the steps and the final product. And we've had a lot of success there, in particular in our evaluations of our scaled up manufacturing for the islet program. We had a process failure. Just as a quick example, that process is 37 days long. And rather than asking the question of, have we mitigated or fixed the problem in manufacturing by making islets and waiting 37 days, we figured out that through our analytics, we could predict the process failure at day 3. And so we cycled all of our R&D and our root cause analysis follow-up on the first 3 days of the process, figured out where we had solved the problem, and then went through and showed that we could make islets successfully. This is just a bit of a snapshot. The core facility keeps growing. All I say is if the cell secretes it, has it on its surface, or contains it, we can measure it. And if there's a tissue, we can do mechanical testing as well. We measure headspace gas for various predictive markers as well. So we have GCMS added to this. So essentially anything that the cell expresses, we can measure it. And I'll just close by saying that we're implementing this in a 30,000 square foot facility in Manchester, New Hampshire. The deep characterization facility is central to that. We have a number of clean rooms. They look empty. This is an old photo. They're now brimming with technologies that we are scaling up. And through the Economic Development Administration, we've now got a grant to expand this to 100,000 square feet across the street. So we have a full campus where we can do basic R&D all the way into final manufacturing for commercial production of living products. I'll also say that we have a number of wraparound capabilities. We have full regulatory quality systems, everything that you will need in a phase appropriate manner in order to be able to push these technologies forward. And we look forward to working together. Thanks.
All right, so thanks everyone for submitting questions. This is the time when we just sort of step through them. I will note in generally you have multiple venues for asking questions. So, I mean, this is one of them. Also, there's a FAQ that will be published, right? So that's, you can always send that in at any time and we will update that routinely. Should we plan on engaging the IV&V? How and how? The scope of the IV&V is dependent on the final solicited work, so you can just hold off on that. But the intention is to test the performance and the reliability of any solution that you guys come up with. We try to kick the tires. This is not an adversarial relationship. I promise you, I've run this 6 times before. People ultimately really end up enjoying working with an IV&V team who helps them think about their technology and what's the most convincing way to establish that your technology is working. But don't worry about it now. For the budget, think about what it would mean, for instance, if somebody comes visit your lab, check out the technology. If you're generating a device, how much would it cost to generate a few devices, send it to an IV&V team. Can we partner with Army, given the role of an IV&V team? So to maintain objectivity in the IV&V process, Tom's group, Army, is not a potential partner for this. However, Army is, not just institutions, they're connected to many people. So he's a great reference to hit up Tom. He probably knows somebody who might have a solution to your problem. So hit him up, and he may be able to connect you with one of the people, institutions, companies that are part of the Army umbrella. The module announcement mentions hormones, cytokines, and other therapeutic molecules, but later it states that only diseases treatable with hormones may be proposed. What is the scope of molecules for the solicitation? So one of the reasons the MAI's draft is that proposals day helps us refine the final one. So thanks for the catch. It is for all of the above. It's not just hormones, right? So anything treatable with hormones, cytokines, and other therapeutic molecules is in scope. What all is needed in the abstract? Should we submit an abstract if we are interested in OTs, or is abstract just for the full proposal pathway? So you don't have to put in an abstract. We really encourage you to provide abstract. Free feedback, right? Feedback that you might get in the full proposal that would have been answered really easily up at the abstract stage. So they provide an opportunity for that feedback. The ultimate vehicle for funding is going to be an OT, right? Abstract submission timeline is very tight from the perspective of forming a collaborative team. Our team changes allowed after the abstract submission deadline. All right, so first thing, we're working on getting through the final approval. It's in the draft form. We're going to give everyone an extra week for proposal abstracts. It seems like this is a recurrent thing, so everyone can enjoy their Thanksgiving and lying around with a full tummy after Thanksgiving. We'll probably keep the final one. Please note, we do not have final approval. It's our new agency. We go through other channels. They're not necessarily working at ARPA-H speed. And so that may be delayed a little bit farther, but don't rely on that, right? Get the proposals abstract in. Okay. How will review consider one technology science team bidding in multiple teams? Yeah, so this happens a lot. It's not a big deal. Like some team has some really great technology and like 3 or 4 other teams want like a part of that technology. Completely cool with that, but obviously you're not going to get paid 4 times the same work. So we just negotiate that after one will have a lead for that, and then we'll work out, how much extra work you need to adapt it for other people's technology. Not a problem at all. Are academic sites allowed? Yes. How many awards are expected? 3 to 5, depends on how much you guys need. Would RAC consider continuous molecular sensors that do not employ living cells? No. Any institutional restrictions? For example, only 1 proposal per university? That's not a thing with us. Can a proposal focus on only 1 TA? No. I mean, we want a complete package. You need to find other people to generate all the capabilities. How soon will we get feedback on abstracts? 1 week after submission. That's our goal, to provide feedback. That could get really complicated in late December, so we'll just see what happens, but we'll try to provide feedback as quickly as possible. So I can say that if we have everything in on the 7th, if I said 7th, we'll have it 1 week after that. Pros, cons of having a small business development company leading a particular effort in our particular leading on participating in a team. So that's fine. We welcome academic performers, large companies, small companies, everything in between. There are no restrictions and no change in our assessment. If pursuing technical tracks 1 and 2, do you need to write 2 technical proposals? So no, you can submit 1 proposal that covers both technical tracks. It's going to be, you're going to have to be efficient with your space. The proposal must make clear it covers both tracks, let us know, and must address the technical details of both tracks. Right. Number 6, 6-page abstract. Okay, so we just gave you some a little more time to submit a proposal abstract. Given many highly effective clinically used first-line cancer therapies today that involve cytotoxic compounds that cannot be produced in mammalian cells, is ARPA-H open to submissions that engineer plant cell lines in the form of ever-living, industrially scalable, highly cost-effective tissue cultures as living pharmacies? So people have other questions of, can we use bacteria? You can use other cell lines if you can keep them alive and viable and not immunogenic in vivo. If you can, I can guess how you might do that with bacteria. If you can do it with a plant cell, that's going to be a very interesting proposal. So, more power to you. Why are MIAs not finalized prior to Proposers Day? This is a little bit Proposers Day. Well, 1 allows us to move quickly. We can put up a draft and then announce Proposers Day to get things moving. The other aspect of is the Proposers Day allows us to refine things. That if we show up at Proposers Day and everyone's going in a different direction, or every time we mention a TA, someone just shakes their head, right? Then it allows us to update. And as noted, some of the dates for final delivery are for the proposal abstract submission are delayed. Is there a specific time for questions, and when will they be answered? Right now. No, sorry, more seriously, the FAQ is rolling. We'll keep that going, until the 4 o'clock on the final submission day, updating those as often as we can for any of your questions. As always, that is publicly available. Please don't ever put in your special sauce, anything proprietary into that, because it goes out to everyone. We have to do that to keep it fair. Yeah, so if you get too close and we get too busy, there is a chance within 10 days. Ask your questions early. That's rarely a problem. Generally, if you're waiting to the very last moment. Anyway, and I already mentioned you probably should put in a proposal abstract. All right, so these FAQs actually they are related. They're publicly available, so I'm not sure we need to dive into that quite so much. And so the next thing is just my wrapping up. Sorry, I don't have the schedule. All right. What are you doing sitting there? Go do so. Once again, thanks for everyone. Thanks for putting the proposers. The lightning talks were awesome. I love them. I glanced at some of the posters. They also look great. Thank you all for coming out. Thank you all for talking. I saw a lot of conversations, a lot of teaming already. That's the whole reason for the day, right? Proposers Day is for the proposers, the team up, come with the best technology possible. So look forward to hearing all of you. I know we'll be talking later today for the for some sidebars, but in general, thanks. Great ideas. I love the community that's already forming around these concepts, and looking forward to working with you all. Thanks.
We're also still accepting teaming profiles if they want to share them.
Okay. And we're still accepting teaming profiles. That's all up online. If you can talk about your technologies, other people for the virtually inclined, so you can go sort through there and find out complementary technologies and capabilities. All right. Thanks, guys. Really appreciate you turning out.